Okay, let's unpack this. For decades, the absolute foundation of modern disease diagnosis, especially when we talk about cancer, has been the histology slide. You know the process. We take a sample, we slice it incredibly thin, and then we look at it under a microscope. Pretty standard stuff. But what if that very first step, the act of slicing the tissue so thin, is actually destroying maybe 95% of the information we desperately need to understand the disease?
It's a huge problem, really, almost a foundational crisis that's been hiding in plain sight for, well, for a very long time. Our source material today is a highly technical paper, but it details a genuine breakthrough that could effectively fix this historical bias. It's called highly multiplexed 3D profiling of cell states and immune niches in human tumors. And it specifically uses a technique called 3D cyclic immunofluorescence, or 3D CyCIF for short.
Right, 3D CyCIF. So that's our mission for this deep dive. Getting into the weeds of this technical paper, we're going to reveal some, honestly, stunning evidence that these conventional 2D tissue sections, they leave hardly any cells intact, almost none. And that severely compromises key biological insights, like figuring out what type of cell you're looking at or how cells are actually interacting. So let's start there. Let's define this massive problem, the flaw in 2D.
Okay, so historically, medicine relies on these really thin sections. But I can typically only 4 to 5 micrometers thick. That's 4 to 5 minutes.
Just to give people a sense of scale, how thin is that, really?
Well, think about a single human hair. That's maybe 70 to 100 micrometers thick. So these medical slices are, 15, 20 times thinner than a hair. Incredibly fine. And this tiny sliver is then used for, well, everything. Your standard H&E staining, the basic look, all the way up to the most cutting-edge spatial proteomics methods people are developing now.
Okay, and this is where the paper drops the bombshell, right? Because it gets really interesting here. The study found that even when they accounted for something practical, like rehydration, the tissue swells a bit when you put it back in liquid, so that 5-millimeter slice becomes maybe 9-milliliter thick. Even then, they discovered that nearly all the cells and, crucially, their nuclei were incomplete, sliced right through.
Yeah, it's not just a minor technical issue. It's, like you said, a fundamental failure of the method we've relied on. The data they present is just.
Sure.
It's a massive correction to decades of pathology. In those standard 5-milliliter resections, the ones used everywhere, fewer than 5% of the cell nuclei were actually intact.
Hold on. Fewer than 5%.
Fewer than 5%. Think about that. We've been trying to understand these complex, three-dimensional biological structures by essentially examining two-dimensional debris. Flat fragments, standard 2D imaging. Just because of the physical act of slicing that thin, it fragments approximately 95% of the cells you're actually trying to study.
That's astonishing, 95% fragmentation. If 95% of the potential information is just garbage, or at least incomplete, what are the real-world consequences? Like, if I'm a pathologist or a researcher looking at a slide, how am I supposed to know if, say, the absence of a key protein signal is real biology or just an artifact because the cell got chalked up?
That's exactly the problem. That lack of ground truth, not knowing if what you see is real, leads to severe consequences. Probably the most notable is what you just touched on, erroneous cell type assignment, or phenotyping as we call it. They found errors in up to 40% of cells. 40%.
40% misidentified. How does that happen?
Well, it happens largely because most analytical software, the tools researchers use to automatically analyze these images, they rely on finding an intact nucleus. The nucleus is like the cell's ID card, right? The software finds the nucleus, defines the cell boundary around it, and then assigns its identity based on the markers it sees. But if that nucleus is mostly chopped away by the thin slice, the software often just discards any signals like protein stains in the cytoplasm that might remain. It can't confidently link them to a cell.
Okay, so the cell might technically be there, or at least a piece of it, with important proteins, but because its nucleus isn't clearly visible in that thin slice, the signal essentially gets thrown in the digital trash bin.
Exactly. Lost data. And the error gets even worse. It's compounded when we look at specific types of proteins. The study found really huge discrepancies, particularly for what are called polarized proteins.
Polarized proteins. What does that mean exactly?
So these are proteins that aren't just spread evenly throughout the cell, like, say, paint on a wall. Instead, they're clustered in specific locations, often in tiny spots called puncta, maybe on one specific side of the cell, or concentrated right at the cell membrane where it touches another cell. Think of immune checkpoints, like LAG3, which they studied.
Okay, so I'm picturing this cell like a complex building, and The polarized protein isn't everywhere. It's maybe just a security keypad right by one specific door.
That's a perfect analogy, exactly. So
Yeah.
if you take just a very thin, random slice through that massive building, what are the chances you actually hit that tiny keypad? Pretty low, right?
Yeah, you'd probably miss it most of the time.
And that's what they found. For these polarized proteins like LAG3 and another one called MX1, they saw false negative rates of 30 to 40%.
30 to 40%. So the protein was actually there, the cell was positive, but the standard 2D slice just completely missed that tiny cluster.
Precisely. It reported it as negative incorrectly. Now contrast that with proteins that are uniformly distributed, sort of everywhere in the cell, like the paint on the wall. For those, like a melanoma marker called RMR-SEM, the false negative rate was much lower, only around 5%, because even a thin slice is likely to catch some of it.
Wow. So the error rate itself depends massively on what you're looking for and where it is inside the cell. The error is baked right into the 2D method itself. And then there's another problem you mentioned earlier, the overlap issue. That must be rampant when you physically squash a dense 3D tumor environment down into a flat 2D image.
Oh, absolutely. It's a huge confounder. When you compress that three-dimensional volume into 2 dimensions, you completely lose the z-axis information, the depth. You can't tell what's in front or behind anymore. So in the paper, they used computers to simulate this. They took their 3D data and created virtual 9-nanometer sections, mimicking traditional analysis. And they saw exactly this problem. There was a T cell, an immune cell, that got incorrectly scored as being positive for an important immune marker called PD-1.
But it wasn't actually positive.
No, it wasn't. What was happening was that this T cell was physically overlapping just along the line of sight, the z-axis, with a completely different cell, a neighboring dendritic cell, which was genuinely PD-1 positive. In the flattened 2D view, their signals merged. 2D simply can't distinguish true physical interaction or co-localization from just accidental alignment due to the projection.
Okay, so the core problem is clear. Thin slicing fragments almost everything, leads to massive errors in cell ID and protein detection, especially for localized signals, and creates false impressions due to overlap. The absolute necessity, then, is depth. This is where 3D CyCIF comes in as the solution. Can you quickly break down the mechanism? Let's start with the cyclic immunofluorescence part. What does that mean?
Right, so CyCIF itself, even the 2D version, is a really powerful method. It lets us measure many different protein markers. In this study, they looked at, I think, 8 to 18 different markers per sample, all in the same piece of tissue. It's called cyclic because you don't stain for everything at once. You stain for maybe 3 or 4 markers. You take detailed images of them, noting their location. Then, and this is the clever part, you use chemicals to basically bleach or inactivate the fluorescent tags on those first antibodies, making them invisible. Then you come in with the next set of antibodies for different markers, stain, image again, and you repeat this cycle, stain, image, strip, until you've measured all the proteins you're interested in, building up a highly multiplexed map.
Okay, so cyclic means repeat rounds of staining and imaging on the same sample. But the 3D element here, that's the real game changer for fixing the fragmentation problem. How do they achieve that depth?
Yeah, the 3D is key. First, they start with much thicker tissue slices. Instead of 4 or 5 millimeters, they're working with sections that are 30 to 50 millimeters thick once rehydrated. So, roughly 8 to 10 times thicker than the standard. These are still prepared from the same standard FFPE blocks used in hospitals, which is important.
So you've got a thick slice, preserving more cells, but how do you see through it clearly? Isn't it blurry?
That's where the second part comes in. They incorporate optical sectioning using high-resolution confocal microscopy. Confocal microscopy is a crucial technique. Unlike a standard microscope that collects light from the entire thickness of the sample at once, a confocal microscope uses pinholes to actively reject light that's out of focus. This allows it to capture incredibly sharp images from just one very thin focal plane within the thick specimen. Then, the microscope moves its focus slightly deeper, takes another sharp image, moves again, takes another. It essentially creates hundreds of these digital optical slices throughout the entire depth of the thick specimen.
I see. So you're not physically slicing anymore, you're optically slicing with light and computation, and then you stack all those digital slices back together.
Exactly. You computationally reconstruct the full 3D volume from all those thin optical planes. Now, instead of maybe 5% intact nuclei, they found something like 80% of nuclei were intact in their 35-meter reference specimen, a massive improvement.
We've genuinely moved from studying, fragmented shadows to visualizing complete cellular architecture. It's like going from a blurry photo of a crowd to a high-res 3D model where you can see every individual. And the resolution they achieved is incredible, down to 140 by 140 nanometers in the XY plane and 280 nanometers in depth Z per voxel per 3D pixel.
It is. That level of detail allows us to study cell structure, protein localization, and interactions within archived human tissue, real patient samples, at a level that was previously really only possible with cells grown artificially in a petri dish.
So just to recap this first part before we dive into the discoveries, we've established that decades of 2D tissue analysis, the standard of care, suffered from a massive, previously underappreciated bias. Because of the thin slicing, about 95% of cells were fragmented.
Right. Leading to potentially huge errors in identifying cells, missing important protein signals, especially those localized ones, and misinterpreting proximity as interaction due to overlap.
And the fix, detailed in this paper, is 3D CyCIF. It uses much thicker tissue sections combined with optical sectioning via confocal microscopy. This preserves the vast majority of cells intact and allows computational reconstruction of the full 3D environment.
Correct. It provides the necessary ground truth by capturing whole cells in their actual spatial context.
Okay, that's the setup. The ability to finally see whole cells in 3D must lead to some absolutely fascinating new biological revelations. What did this 3D view actually reveal about how tumors and immune cells organize themselves? What new things did they see?
This is really where the power of the technique shines, uncovering biology that was simply invisible before. First off, just the basic architecture. The cell packing is incredibly dense. In the melanoma samples they studied, and the surrounding supported tissue, the stroma neighboring cell membranes are often only about 1 micrometer apart. It's really crowded.
A single micrometer. That's incredibly close.
Very close. And related to that, the actual space available for the cell's machinery to work the cytoplasm, the space between the outer cell membrane and the nuclear membrane, is surprisingly shallow, often just 1 to 6 months deep.
So the environment is like a tightly packed, crowded mosh pit, and the individual cells are quite thin in places. That density must make interactions complex.
It does. And what really struck me, the thing that 2D was utterly missing, was the visualization of these incredibly fine, extended structures that cells use to communicate and explore. It's like seeing the hidden communications network, the cellular equivalent of maybe telephone wires or probing antennas.
You mean things like filopodia, those thin extensions?
Exactly. Filopodia, dendrites, membrane ruffles. 3D imaging was absolutely essential to see these. They are highly extended cytoplasmic processes, often very long and thin. In a traditional 2D slice, these would just appear as meaningless, disconnected specks, if they were captured at all. You'd have no idea they were part of a larger functional structure reaching out from a cell.
Give us an example from the paper. How significant are these structures when you see them properly in 3D?
Well, take the immune cells, which are critical in cancer. They saw CD8 plus T cells. These are the main killer cells of the immune system, extending multiple filopodia, sometimes longer than 5 or even 10 nanometers, out from their main cell body, probing their environment. Even more impressive were the dendritic cells. These are like the generals of the immune system coordinating the attack. In 3D, these dendritic cells extended massive filopodia and complex membrane ruffles, reaching out 20 to 30 micrometers.
20 to 30 micrometers. That's huge compared to the cell body. What were they doing?
They were specifically using these incredibly long extensions to make contact with multiple T cells simultaneously. The researchers proposed this specialization, this ability to reach out and touch many partners, is the physical mechanism that enables dendritic cells to switch from just sampling their environment for danger signals, antigens, to actively presenting those signals to T cells and initiating an immune response.
Wow. So the immune system is literally reaching out, communicating via these complex physical extensions that are just completely invisible in the standard 2D clinical view we've relied on. Amazing.
I also recall reading in the paper that 3D imaging corrected a major misconception about cell shape, and it turned out to be just a matter of perspective, of geometry. Yes, that was a fascinating example in melanoma. Based on 2D images of melanoma invading the skin, specifically vertical growth phase or VGP melanoma, researchers had previously concluded that the cancer cells fundamentally changed their shape. They looked kind of round when they were deeper in the tumor, but appeared elongated or stretched out at the invading margin. People thought this was an intrinsic change in cell biology related to invasion.
But it wasn't.
No. When they looked in 3D, they proved it wasn't a change in the cell's actual shape at all. It was simply a difference in orientation. The cells were basically the same shape, but at the margin they happened to be rotated about 90 degrees compared to how they were oriented deeper down. So in 2D, you were seeing them side-on versus end-on.
Huh. So it's like looking at a cigar. End on, it looks round. Side on, it looks long. The cigar didn't change shape, just your viewpoint did.
Precisely. Without the 3D context, you might incorrectly assume a fundamental change in cellular identity or behavior, which wasn't actually happening. It's proof that sometimes seeing the whole picture changes everything.
That perspective shift is powerful. This whole cell view also allowed them to capture fleeting, really dynamic events in action, didn't it? Like immune cells actually caught in the act of moving between tissues.
Yes, they managed to capture snapshots, providing clear physical evidence of transendothelial migration, which is also called diapedesis. That's the process where immune cells squeeze out of blood vessels to enter surrounding tissues.
What did that look like in 3D?
Well, they saw B cells, another type of immune cell, physically flattened against the inner wall of a blood vessel. That morphology is classic for cells preparing to migrate out. But even more strikingly, in one incredible 3D snapshot, they captured a B cell with its nucleus, the main part of the cell, actively traversing the vessel wall it was partway through, while the rest of its cell body, the cytoplasm, was still inside the vessel lumen.
Wow, caught mid-transit.
Exactly. A highly complex, dynamic action that happens relatively quickly. Something that would be completely obliterated, just erased, by the random chance of a thin 2D slice hitting it at the right nanosecond. You'd never recapture that complexity.
Okay, so we've gone from basic architecture and cell shape to seeing these dynamic cellular processes. Now, moving deeper inside the cell itself, 3D imaging even allowed them to characterize tiny functional differences within the cell, right? Even in these archived, preserved human tissues.
Yes, that's another major advantage. We're talking about visualizing detailed organelle structures, things usually studied only in fresh or cultured cells. For instance, they could clearly see the highly characteristic multi-lobed nucleus shape that defines neutrophils, a type of immune cell. They could also identify distinct peroxisomes, small organelles involved in a metabolism, by staining for a protein called catalase, and they could map the precise subcellular localization of key functional proteins. Take that immune checkpoint protein, LAG3 we mentioned earlier. In 3D, they saw it wasn't just generally on the cell, it was often clustered in specific puncta, maybe 1 to 20 distinct spots per cell located both inside the cell and right at the plasma membrane, likely where it's functionally active.
And you mentioned earlier that the location of a protein could indicate the cell's functional state.
Exactly. They showed this beautifully with granzyme B, or GZMB. This is the main weapon, the executioner protein, used by cytotoxic T cells to kill targets like cancer cells. When they looked at CD4 plus T cells, which are more like helper or memory cells, the GZMB protein was present, but it was kind of diffuse, spread out, maybe globular inside the cell, consistent with an activated memory state perhaps, but not immediate killing. But when they looked in CD8 plus T cells, the actual killers of the GZMB was strictly punctate. It was neatly packaged into distinct granules, like little bombs, ready for immediate deployment towards a target cell upon contact.
So the 3D view reveals not just if a protein is there, but how it's organized, which tells you about the cell's immediate function, presence versus readiness.
Precisely. It adds a crucial layer of functional context.
Okay, this is really powerful. Let's do another quick recap here. We've seen that 3D CyCIF moves beyond just fixing the fragmentation issue. It reveals the true, dense architecture of the tumor microenvironment.
Right. And it crucially visualizes the previously hidden network of cellular extensions, filopodia, dendrites essential for communication and probing.
It corrects misinterpretations about cell shape that arose purely from the limited 2D perspective, and it allows us to capture dynamic events like cells migrating across vessel walls.
And finally, it brings subcellular detail, visualizing organelles and the functional localization of proteins like LEG3 and granzyme B, even in archived human tissues, revealing not just presence, but functional state.
Okay, we have intact cells, we see their shape, their extensions, their movements, even their internal organization. Now, let's shift focus to maybe the most critical aspect for understanding cancer and immunity, the language of cell communication itself. How does this high-resolution 3D CyCIF allow us to map the functional immune niches, the specific neighborhoods where key interactions are happening, especially in the densest parts of the tumor?
This is where that nanoscale resolution really becomes critical. The precision allows them to move beyond just saying cells are near each other to actually detecting functional contacts, often visible as specialized structures or protein clusters right at the membrane interface. A key example is juxtacrine signaling complexes. Remember, juxtacrine means signaling that requires direct physical touch between cells. They looked at the immune receptor PD-1 on T cells and its ligand PD-L1, often found in tumor cells or other immune cells, which acts as a brake on the T cell response. In 3D, they can see PD-1 and PD-L1 molecules clustering together into distinct focal points, or puncta, specifically and only at the interface where a PD-1 positive T cell was physically touching a PD-L1 expressing cell, like a dendritic cell in their example.
So they could actually see the molecular handshake forming.
Exactly. They could even count these functional contacts. In one case, they found 13 distinct PD-1, PD-L1 foci concentrated over a tiny contact area of just 40 square micrometers between two cells. This confirms it's not just proximity, it's a functional signaling nexus being formed.
That level of precision, seeing the actual molecular interactions, allowed them to do something truly novel, didn't it? They proposed classifying different types of cell interactions based on the actual physical spacing between the membranes, moving beyond just touching or not touching.
Yes, this for me is one of the most significant conceptual advances in the paper. Based on the precise 3D distances, they defined three distinct types of membrane-to-membrane interactions. First, they identified type I, direct binding interactions. This is characterized by tight pixel-level overlap in their high-resolution images. This indicates an incredibly small physical gap between the cell membranes, estimated at around 70 nanometers.
70 nanometers, let's put that in perspective again. A human hair is maybe 70,000 nanometers. The old thin slices were 5,000 nanometers. We're talking incredibly close.
Exactly. Nanoscale precision. And that 70 millimeter spacing is biologically significant. It's the precise distance consistent with the formation of an immunological synapse. That's the specialized tight junction T cells form when they recognize their target and get activated,
right,
or deliver a killing blow. It's the critical handshake.
Okay, so type I is the direct functional handshake, the synapse, leading to activation, killing, or strong signaling. What about type 2?
Type 2 membrane apposition involves extensive contact area between cells. They're clearly touching over a broad surface, but without that super tight pixel-level overlap. The measured gap here is slightly wider, maybe 300 to 600 nanometers. It's still very close, but not synaptic. The researchers speculate that this kind of close apposition might be structurally ideal for a different kind of communication, paracrine signaling.
Paracrine, meaning cells releasing signaling molecules like cytokines that act on their immediate neighbors across that small gap.
Exactly. Facilitating local chemical communication rather than direct binding signal, sort of like talking across a very narrow alleyway instead of shaking hands.
Makes sense. And then there was type 3, which sounded more like a neighborhood gathering. They mentioned complex immune cell clusters they called lymphonettes.
Right. Type 3 neighborhood clustering involves looser packing. The cells are still relatively close, with membrane separations around 500 nanometers or so, but the contact area is typically smaller, maybe just 1 to 2 square micrometers. What they observed were these complex networks or clusters involving multiple immune cell types, T cells, B cells, dendritic cells, all loosely interacting in these lymphonettes. They provocatively suggest these might represent the very earliest stages of formation for something called tertiary lymphoid structures, or TLSs.
And TLSs are important because...
Well, the presence of mature TLSs within tumors is increasingly recognized as being strongly correlated with a better patient response to immunotherapy, particularly checkpoint inhibitors. So being able to spatially define and perhaps quantify the earliest formation, these nascent type 3 lymphonettes, could be really important clinically.
This detailed classification, type I synapse, type 2 apposition, type 3 clustering it, really drives home the point that a single immune cell isn't just doing one thing, is it? It's involved in complex simultaneous interactions.
Absolutely. The ultimate spatial precision showed exactly that, what they call multivalent immune regulation. They provided a stunning example of a single cytotoxic T cell, GZMB plus CD8 plus, caught in the middle of multiple, potentially conflicting signals at the exact same time. So this one T cell might be, 1, polarized towards a tumor cell, presumably forming a type I synapse ready to kill. 2, simultaneously being enveloped by long philopodia extending from a nearby CD4 plus helper T cell, maybe receiving help signals. And 3, yet another distinct contact point, potentially being inhibited or regulated by a PD-L1 expressing myeloid cell through a type 2 or maybe even type I interaction.
Wow. So it's getting go signals, help signals, and stop signals all at once from different neighbors, mediated by different types of physical contact, all happening in a tiny space.
Precisely. It's this incredibly complex, multi-layered regulatory conversation happening simultaneously around a single cell, within a volume smaller than the thickness of a traditional 2D slice that would have averaged or missed it all.
That complexity is staggering. And beyond just the geography of interaction, this deep 3D profiling also revealed insights into the cells themselves, particularly the flexibility or maybe the plasticity of T cell states, right?
Yes, exactly. Because they could measure 10 different lineage markers on T cells simultaneously in 3D, they could get a much more nuanced picture of T cell identity. And what they found were overlapping transitional states. T cell identity wasn't always clear-cut. Cells existed on a spectrum. A key example involved a specific subset called progenitor-exhausted T cells, or TPEC-X cells. These are CD8 plus 2 cells that express markers like TCF1, PD1, and LAG3.
And TPEC cells are important because they're thought to be the ones that can be reawakened by checkpoint inhibitor immunotherapy and actually drive the anti-tumor response.
That's the current thinking, yes. They are critical for therapy success. And what the 3D profiling showed was that these TPEC cells weren't a distinct, isolated population. They found cells with hybrid phenotypes overlapping significantly with traditional resonant memory T cells, TMEM, and effector T cells, TEFF. They even identified populations they called TPOP that seemed to bridge these states.
So T cell identity isn't fixed. It seems much more fluid, maybe constantly shifting based on the complex signals and interactions happening in that 3D niche we just discussed.
It strongly suggests that yes, the local 3D context likely plays a huge role in dynamically shaping T cell fate and function.
Okay, and one final major finding related to mapping function to location. This 3D approach allowed them to pinpoint the very beginning of cancer spread, connecting inflammation directly to the act of invasion.
That's right. Within the melanoma samples, specifically in an area called the melanoma in situ MIS region, they found these very distinct localized domains, maybe 50 to 100 micrometers in diameter. These small niches were characterized by highly active interferon signaling a key inflammatory pathway. They could see elevated levels of proteins like IRF1, MX1, and MHC class I, all indicators of interferon activity.
So little hotspots of inflammation.
Exactly. But crucially, the 3D mapping showed that these interferon-positive inflammatory niches were the precise physical locations where the malignant melanocytic cells were observed, crossing the boundary between the epidermis and the dermal epidermal junction, or DEJ. And at that exact spot, the invading cancer cells were making contact with immune cells within the niche.
Ah, so it provides a direct spatial link. The inflammation isn't just general, it's happening right where invasion starts, suggesting a direct interplay that was previously just inferred but now visualized.
Precisely, a connection between inflammation and the very first step of invasion, verified spatially in 3D human tissue.
Okay, so let's try to synthesize this. What does this all mean? We've gone through the flaws of 2D and the solution of 3D CyCIF, the new biology it reveals, and the detailed mapping of interactions. What's the big picture takeaway?
The big picture is that 3D CyCIF and similar high-plex 3D imaging methods are incredibly powerful tools. They aren't necessarily meant to completely replace standard 2D histology for all diagnostic tasks tomorrow, but they provide the essential ground truth data. They allow us to finally understand and, importantly, correct for the massive 95% fragmentation bias that has unknowingly plagued thin section analysis for decades. It's like getting the answer key to check our previous work.
So it's a reference standard, a way to calibrate our understanding.
Exactly. And more than that, this new capability to study molecular events like PD1, PDL1 clustering, right down to the level of specific membrane contact types, the type I and 2 and 3 interactions, it fundamentally changes how we need to model and understand the tumor microenvironment. Proximity isn't enough. The type of contact matters.
We really need to think in terms of nanometers and interaction types, not just microns and neighborhoods.
That's the shift in thinking required, yes. If we truly want to understand why an immune cell is active or suppressed, or why a therapy works or fails, we need to know if it's forming a tight 70 nanometer type I synapse, or if it's just sitting 500 nanometers away in a type 3 cluster, receiving different signals. They imply vastly different biology.
And we've seen compelling evidence here that the specific T cells thought to be crucial for checkpoint inhibitor response, those TPEC cells, are deeply embedded in these complex multivalent interactions. They're receiving multiple signals at once.
Right. Immunotherapy effectiveness seems to rely entirely on manipulating these precise nanoscale contacts and signals within the tumor niche.
Given that, relying on data derived from methods that, as we established, fragment 95% of those cells and miss the crucial interaction details.
Well, it seems scientifically unsustainable as we move forward, doesn't it? It certainly highlights a major gap between the complexity of the biology and the limitations of our previous tools. 3D spatial profiling offers a way to bridge that gap.
Which brings us to a really important final provocative thought for you, the listener, to mull over. If the success or failure of cutting-edge cancer immunotherapies truly hinges on the actual geometry and functional proximity, those nanoscale type I contacts versus type 2 or 3 interactions happening within a specific patient's tumor niche, then how quickly must clinical research and maybe even diagnostics move to integrate this kind of deep 3D spatial profiling? How fast do we need to shift towards personalizing treatment based not just on bulk measurements, but on the actual spatial reality of the cellular conversations happening inside the tumor?
That's the critical question, isn't it? It's a fascinating challenge at the intersection of technology, biology, and clinical translation. Definitely something to watch closely.